You there. Um, as usual, if you have any questions on the disease stuff, homework that you did last lesson, then please let me know. We have questions. As a reminder for those of you that, if anybody's new, the odd people might be. First 45 minutes or so, I will do Key Stage 3 stuff, year 7, 8, 9. And then we will do the GCSE stuff, even though people in year 10 also did this topic. Um, hello. Can we do, yes, hi. Question 6, 7, 9 in booklet one. Yes, so if you guys are specifying which booklet you're doing, just say one or two or three. I have 1, 2, and 3 ready to explain. I'm Fareed. Hello, Fareed. How are you? Long time no see. Um, right, I'm going to start doing questions. Um, so 6, 7, and 9. Yes, that the booklet one that you guys had was a bit odd, isn't it? Quite a lot of the stuff that you did in the lesson wouldn't have been directly explained for the homework, just because it's a little bit odd. But the stuff you did in the lesson is still very important. Um, 6, 7, 9. Oh, yes, 7. So question 6 is this one. Okay, which part of a cell passes on information is the question. The part of a cell that passes on information is the nucleus, or you could say what's inside the nucleus, which is the genetic material. All of your information in biology is inside your nucleus. It's your genetic material. It's all inside there. Then, part B. MRSA bacteria can cause serious infections in people who are ill in hospital. The bacteria can live on a healthy person's skin or in their lungs without causing any harm. Use this information to fill in the below. Two ways it can be spread from person to person and how you can prevent it for each method. So you could mention that the most obvious way of spreading a disease is by coughing or sneezing. Um, coughing, sneezing, right? That would be that. And how could you prevent getting ill from coughing or sneezing? You could wear a mask or you could cover your mouth or your nose. Right, mask or cover your mouth or nose in some particular way. That would work. Um, in that sense, the other major way of spreading disease is if you have something on your hands and you touch someone else and you pass it on by touch. How could you prevent diseases spreading from your skin? By washing your hands regularly or using hand sanitizer if you like. And there you go. Right, that's that. Um, This is booklet number 1. That's what this is. Right. How does a vaccination prevent people getting ill? The things that you want to say is any vaccine, when you get that injection, contains a dead or weak version of the virus. Okay. So a vaccine contains a dead or weak version of a virus. Right, then what happens is your white blood cells, in response to that, they think it's the real disease. And so they produce these things called antibodies to try and destroy it. Okay, and crucially, if you get the real virus in your body, then the it can be quickly destroyed because we have seen it before. We know which antibody kills it. These antibodies, think of them as little soldiers that can kill specific diseases, but you need to know which ones work with which disease. But once you've seen it once, then you can produce them quickly. So you can say that if the real virus enters, it is quickly destroyed. And that should be the answer to quite a lot of questions across the three booklets because vaccinations are everywhere. Okay, done. God, loads of questions here. Yeah, I'm coming to those booklet one. Yeah, right. Yeah, yeah, yeah. Booklet. So if you're doing year eight, then do booklet two. I thought we were doing maths. You shouldn't have done. Let me check. When was your lesson? No, in the last lesson that you did, you must have done biology. All of you must have done. Yeah. Um. Yeah. I should also let you all know, since you're all here, that you will get an email tomorrow to let you know that at the end of this month you will have a test, which I assume some people have been told already. Um. Six, seven, and nine. Right, question seven. The dentist leaflet shows how plaque can cause tooth decay. It gives you some information here that you meant to read and understand. Anybody can answer this question correctly if you read and understand the question carefully. Because it tells you that plaque is this stuff that forms on your teeth. Hopefully you don't have any. But bacteria live inside it, so they're kind of protected by it. And then bacteria use sugar every time you eat some sweets to produce some acid, and that acid decays your teeth. So if you have plaque, then it's very difficult to get rid of this bacteria. If it's difficult to get rid of this bacteria, then it's going to cause tooth decay because it will keep producing acid. Explain how reducing the amount of plaque can reduce tooth decay. So if you reduce the amount of plaque, you will reduce the amount of bacteria. So there will be less bacteria because they've got nowhere to hide. And if there's less bacteria, what do the bacteria do? They produce acid. So there'll be less acid produced. That is how you answer these questions. You did not need to have learned that to answer this question correctly. You just need to read the information you get given and then apply it. Yeah, I'll get to all of the booklet questions in just a moment. I just need to get through these three from this one, and then I will move on. I'll do it in order that's been asked. So six, seven, and nine I need to do from here. Using an alkaline toothpaste reduces tooth decay. Give a reason for this because acids cause tooth decay. What does alkali do? Alkali neutralizes the acid. That's why toothpaste usually contain an alkali because it gets rid of the acid. Um, A group of boys wanted to find out how well plaque is removed by brushing teeth. Every day before they brush their teeth, the boys chewed a tablet that stains plaque red. Explain why the boys looked at their teeth before and after brushing, because they were trying to compare. That's why you do things before and after. You compare the amount of plaque before and after, because they'd be able to see it if it was there, because it'd be red. Okay. Teeth with plaque stain. Let me see if anybody's asking any relevant questions for now. No, not yet. Um, okay. See, this one here is a weird one. It says the boys used a grid drawn on clear plastic to measure the area of the plaque on their teeth, and B is better than A. Why is B better than A? It's basically because it's more precise, because you see what I mean? Here you count one, two, three, four, five, six, but then it's not really, it's counting loads of bits that's empty. You're going to be more precise by having a smaller grid. So more precise is what you want to say. Um, the squares on grid B represent 1 millimeter squared. Use grid B to estimate the area of the tooth covered by plaque. So we've got to do some counting. So I would do it roughly like this. One, two, three, four, five, six. That makes six. So that's counted. And then that and that roughly is 7, 8, 9, 10, 11, 12, maybe 13, 13, 14 roughly. Let's see what the answer says. Anywhere from 13 to 17 is a correct answer. Okay. And then we also had question nine, which I will do. Jane stored some milk at room temperature for 5 days in a sealed container. She uses a pH sensor, remember that detects acids and alkalis, to record the pH of the milk for 5 days. Just on this note, just for everybody to understand, milk contains, well, it contains fat. Okay, that's what milk contains. And Over time, what happens is that fat gets broken down into these things called fatty acids, which you may recall from when you did digestion. Um, and that makes milk acidic. That's why you don't really like having it after a while. And that's what's happening. You can see the pH over time is going down. If it's going down, it's becoming more acidic. Okay, let's see what happens here. Jane predicted the number of live bacteria in the milk would change as shown below, which is one reason why the number of live bacteria would start to decrease after 3 days. Right, it's a difficult answer, but bacteria don't like it when it gets too acidic. That's why you have this acid in your stomach. And so the amount of bacteria will start to decrease after 3 days because after 3 days, the milk starts to get very acidic, as you can see here. So it is too acidic. Okay, what evidence from graph one suggests that there were still some live bacteria in the milk on day 5? Um, well, the graph's not at the bottom, is it? Um, is that the answer? Part two. From graph one suggests that there were still some live bacteria. Oh, it's because the graph is still going down. What the bacteria are doing are um, feeding off the milk and turning the fat into acids. So, because the graph is quite a hard question, but it's the answer is that the graph was still going down. Right. All right, and then last couple of questions on this. On Jane put some fresh milk in a sealed container in the fridge. She measured the pH of the milk every day for 5 days. Draw a line to show the pH of the refrigerated milk after for 5 days. Um, so basically, if it's refrigerated, it won't decay as quickly. So you just need to draw a line that still starts here but finishes higher than the other line. Okay, because the whole point of a fridge is that it helps your milk last longer. Um, and on graph 4, draw a line for how the number of live bacteria will change over 5 days. Um, that's quite a hard question as well. It's going to be like that, basically, because it'll just take longer for the same thing to happen. That's the idea of it. Right, that's quite a hard question. Let me move on to any other questions that have been asked from other booklets. An STD. I wasn't on my lesson and I've seen it in my notes. A sexually transmitted disease is an STD. Wait, which booklet is this? Sarah, I thought you were doing maths. 1C, somebody's asked. Is that from this booklet? 1C. Just make sure you specify. Oh, booklet one. Yes, 1C. Um, Let me just do that one first quickly. The relationship between the variables from 30 to 90. So 30 to 90. All you need to do is explain what is happening here. You need to explain as the temperature increases, the volume of the dough increases up to 60 degrees. Then after 60 degrees, the volume decreases. That's how you have to explain, say what you see, basically. So what's happening on this graph is that as the temperature is going up left to right, the volume is going up. That's why this is this bit of the graph. Then after this bit, as the temperature goes up, the volume starts to go down. That's what you're supposed to do. Right, I think booklet 3 seems to be popular here. Okay, 6C, booklet 3. It's going to be a big one, isn't it? I'm not answering this question. Explain how a vaccine makes someone immune. There is absolutely no chance that you do not have details of how a vaccine makes you immune in your notes. So I'm not going to answer that question. Um, right. So also I did explain it earlier. There is a process that for vaccines, the answer is always the same. It's all about a dead version, and then your white blood cells producing antibodies, and then your next time, if you get the real disease, your white blood cells can quickly produce those antibodies, which means you don't actually suffer from an illness more than once. Right, let me see what else we've got here. So 1D, year 9, 1D, where are we? Okay, right, this one's a little bit harder, fair enough. Question one says quite a lot of information here. 1D. Right, 1D. That's this one, isn't it? Quite a few people have asked for 1D. Okay, because presumably because it's long. Um, right. A scientist investigated the effect of three different antibiotics on three different types of bacteria. They grew some bacteria on plate A and then some different bacteria on B and a different bacteria on C. They used three different antibiotics. Okay, so one, two, and three. And then they left all of these for a while and came back and had a look at what happened. So where you see white circles or white space, that is where the bacteria has been killed. So we can see just by looking at this that for bacteria A, would you agree that this antibiotic number three is the best one at killing it? That's why, because it's got a bigger circle. Whereas for bacteria B, they're all pretty good, but one is the best because it's got the biggest circle around it. That means it's killed the most of that bacteria. So for bacteria B, I'd be better off using this number one antibiotic. Two and three would work, but they wouldn't be as good as number one. For bacteria C, I would definitely use bacteria two. Sorry, antibiotic two, because antibiotic two kills the most of this bacteria. One kills some of it, three doesn't kill any of it. So, what's the question? Compare the effectiveness of the three antibiotics at killing the different types of bacteria. So, I'd explain that antibiotic one kills a little bit of bacteria A, but it's much better against bacteria B and C. It's the best against B, so I would use antibiotic one for bacteria B. Antibiotic number two does kill some of A and B. You can see that there, it's killing some of them, but it kills a lot of bacteria C. So we would generally use antibiotic two against bacteria C. And number three works on A and B. It doesn't work at all on C. So we would not use antibiotic three at all if you had bacteria C. You would probably use it most likely on bacteria A. So really, that's what you do. All the words I just said are the words that we use. Is the medicine that kills viruses antiviral? I think there is a medicine that can treat certain viruses. It's called an antiretroviral. There you go. This is not booklet one anymore. This is booklet three, which I've done the questions on booklet one. I think that I'm going to be doing. Um, right. I'm doing them in as much order I can. So 1D, loads of people seem to ask 1D, or at least the same person asked for 1D lots of times. Um, if you weren't in the lesson and haven't got the homework booklet, then hopefully it'll be with you soon. Bear in mind that if your lesson's like on a Sunday, then sometimes it will go out on Monday night or Tuesday. It won't get to you in time. But you should have received it electronically. That's why we do send it electronically. Um, 1E booklet 2, I will come to that in a moment. I'm just checking if I've done all the booklet 3. 1D, 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 everybody saying 1D. Right, I've done 1D, haven't I? Okay. 11B and C booklet 1. I might come back to that later. I won't be coming back to that now because I did booklet 1 to begin with. Right, booklet 2. There are some questions. Any more from booklet 3? 1C, booklet 3. Let me see what the answer says for that one, actually. It's a good question. I think wash hands would be fine. Generally, to stop bacteria spreading, you can wash your hands. That would be that one. Um, 1C, booklet 3 done, 1C done. Right, I'm moving on to booklet 2. I feel like I've done everything that anybody asked me from booklet 3. Booklet 2. 1E seems to have been asked. 1E. Let's do 1E. Again, I'm not doing 1E because if that is not in your notes, then I'm going to be more than shocked. How a vaccination can prevent an illness is 100% in your notes. Use your notes. 6C, booklet 3. Same again. I'm not answering that because it's in your notes. There is absolutely no chance I'm answering a question again about how a vaccine works because there is no chance it is not in your notes. So you should be able to use your notes to answer any question about how a vaccine works. Right, fine, not doing that. That's it then, really, isn't it? I'll go back to booklet one. 11 B and C, booklet one. Okay. Um, Spots may be caused by a bacteria. A researcher affected, right? Okay. They placed two small discs on the surface of the jelly. One disc had been soaked in spot lotion, which presumably kills some bacteria. The other disc had been soaked in water. Right. So, what this is showing here is that this spot lotion kills some of this bacteria. That's why it leaves a white circle around it. What had happened to the bacteria in the clear area? It had been killed. That's what you want to say there. Um, what was the control in the experiment? The control is the disc soaked in water. A control in an experiment is kind of one bit of it to which you don't do anything. So it allows a comparison. It basically shows that, oh, look, nothing happened around here, but it did happen around here because this one was soaked in spot lotion and that killed the bacteria. Um, to see booklet two, let's have a look what that is. This is a vaccine question again. In school laboratories, bacteria should be growing at 25 degrees. One reason why companies testing new antibiotics grow bacteria at 37. What number? When you see 37 degrees C, there's one thing that should jump out at you immediately. There's just one thing that the number 37 for temperature is associated with. It is your body temperature. And so, yes, that's why if you're testing antibiotics on bacteria, you kind of want to do it at 37 degrees C because you're trying to replicate what happens inside the body. So that's the temperature we use. Okay. In a school, it's different because you're not trying to, cure things. You're just trying to demonstrate it to some students. So we wouldn't do it at a high temperature in a school. And also it can be quite dangerous to do it at too high a temperature. Um, 7E, booklet three. Let's have a look at what that says. Um, Use of mosquito nets has helped reduce the number of deaths from malaria each year. Other reason for the reduced number of deaths from malaria each year. Malaria is an infection that is spread by mosquitoes biting one person and then biting another person. And that the biting process transfers the malaria from one person to another. So one way that you could reduce the number of deaths from malaria could be, and without looking at the answer, I'm just going to say it. I suspect it's using insect repellent. So, insect repellent, that stuff that you can spray on your skin, that generally slows down. It prevents you from being bitten. If you can not be bitten, then you wouldn't get the disease because the only way to get the disease is to get bitten by a mosquito. Um, oh God, 6D, booklet three. Is that what I just did? No, 6D. A child who has not been given mumps can't why not antibiotics, presumably because mumps is a virus. Antibiotics only kill bacteria. This is a fundamentally important thing that should have been highlighted to you. Is that antibiotics only affect bacteria. They only kill bacteria. They do not do anything else. 7F, booklet three. I'll move on. I'll make sure I answer every question, so don't worry. 7F again. That's a harder question. Again, I really don't want to do these questions. I'll tell you, the only reason I don't want to is because I know what you are taught in every lesson, and I know what is in your notes. And there is absolutely no way that this isn't in your notes. So I'm not going to write anything down. I'm going to say things. Um, how does the human body prevent pathogens from entering? Um, One way in which it prevents things from entering is that your skin has a barrier, is a barrier that stops pathogens getting into your blood. You have mucus, the snot in your nose, and hairs in your nose that trap things that you might breathe in. That stops them from entering. You have hydrochloric acid in your stomach that tries to kill bacteria to stop it from going any further into your body. That's that. Then we move on to the next bullet point. How does the body defend itself against pathogens inside the body? You have white blood cells. The way they protect you from disease is three main ways. They can engulf and consume pathogens. That's like when they eat them. They can produce these things called antitoxins that counter the toxins produced by bacteria, and they can produce antibodies. I'm not writing this down deliberately because there is very little chance that this is not in your notes. This video will hopefully end up on YouTube. It is currently streaming. And therefore it should automatically end up there. Are you sure? Um, yeah, it should do. Yes, should do. But anyway, the first thing to remember when you guys do your homework, right? Look at your notes because many, I'm not saying all, many of the questions will definitely be. Those of you who have asked me about a vaccination today, there is no chance that that is not in your notes. Do white blood cells produce antitoxins? Yes, they do. That is correct. Um, Let me see what else we've got here. I'll work my way up. 5B booklet 2. Hmm, a vaccine. No answering that. I'll answer the second part. Yeah, second sentence. Sorry, yeah, second sentence. MMR. This wouldn't have been in your lesson because I know this. I know these things. MMR vaccine is made of three diseases: measles, mumps, and something called rubella. Okay, that's what it is. Okay. Um, 2 and 4, booklet 2. Yeah, any particular part of it? Because if you just ask me question 2, then I'm not going to answer it. Question 4, I'm not answering that. This is ticking a few boxes. Um, yeah, I'm going to need some specifics there. No, I'm not doing all of it. Da, 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 da. Booklet 1, question 11D. I think I already did that, but I'll show you the answer. Oh, I didn't do that. 2 safety precautions you could take to avoid contact with the bacteria. You could wear gloves, um, and you could actually. What's the other answer? Um, you could wear gloves, or you could use tweezers as well when you're handling things. You can wear a mask, that sort of stuff. Um, do white blood cells produce antitoxins? Yes, they do. Right. Okay, I'm going to try to remove some things now as I go here. Um, right, I'm going back to the top. Just give me a moment while I explain how MRSA strains of bacteria became difficult to treat. That's quite a hard question, actually. I'll tell you that. So the thing about bacteria is that it can constantly change, and by change we mean mutate. Okay, so it can mutate, and if it mutates, it can become resistant to antibiotics. So some bacteria can change, and so antibiotics don't kill them, and that makes it difficult to treat, right? Because our main treatment for bacterial diseases is antibiotics. But if some of those bacteria change in some way and become stronger, then it becomes harder to treat them. Okay, right, that was that. Let me see where we are. 7D, booklet 3. So many questions today. Um, Predict the number of people who died from malaria in 2017 if the trend stays the same. And I was going, it's just prediction for 2017. Look what's happening here. From 2013 to 2015, it dropped by like 2. So in the next 2 years, it might drop by like 1. So like 91. That'd be my prediction. Without looking at the answer. Okay, I really need to change the setup of these lessons. Um, but booklet 1 to be part 2. Did I do that one? I just want to make sure I've done that one. That's a good question. Give one reason why it's important for Michael's health that the first aider. So if somebody fell over, cut themselves. Why is it important that the person who's dealing with them, the first aider, wears rubber gloves? Because you want to avoid passing on any infection into that person from your skin. So you want to wear rubber gloves to avoid passing anything on to Mike. Because now that he's cut himself, you could easily pass something into his body through his cut. So you want to wear gloves to avoid doing that. That was that. I think I did that one. 7D, I think I did in booklet 3. I'm just going to go in order now and just make sure everything's done. Did 7D. Did that one. Yeah, last time you couldn't find it on YouTube mainly because I didn't turn it on. I think that's what happened. That's the mistake I made on New Year's Day. Um, um, 7D, I think I've done now, haven't I? Part two, booklet 2. Is that the question that somebody's asked me? Part 2. Um, that I'm not an answer that. It's a graph. 6AB booklet 3. So yes, this MMR stands for measles, mumps, and rubella. So you can figure that out from there. Mumps vaccines contain mumps viruses. Why do the viruses not cause mumps? You know that in a vaccine, it contains a dead version of a virus, so it doesn't actually make you ill. Done that. Booklet 1, question 1C. Did I do that one? Um, Yeah, so I explained here that when you are doing a question with a graph like this, you just have to say what you see. So describe the relationship between the variable from 30 to 90. That means explain what happened here and then what happened here. So say that as the temperature increased, the volume of dough increased up to here, 60. After 60, the volume decreased. That would be fine. Um, can you tell me what is biodiversity? I shall tell you. But actually, biodiversity, I believe, is the variety of different plants and animals species on Earth, or the variety of different species on Earth. We'll come to that in a moment. Um, just want to make sure I've done everything. B2 question. Have I done everything? Anybody got any more questions? Booklet 2, C part 2. B2 question. I don't know what that means. B2 question 2. Booklet 2, C part 2. That also doesn't mean anything. Um, you have to tell me the actual question number. C part 2 doesn't tell me. Do you mean question 2? Question 4? You know, 4. Um, Not in the last question, you didn't. 4. 4C part 2. Okay. This one. What would a doctor give to a patient to relieve the symptoms of flu? Painkiller would probably work. Painkillers help to relieve the symptoms, but they don't actually get rid of the flu. They just help you, like, if you got a headache or whatever, like paracetamol, these things. Um, Right, I think that's pretty much it. As I say, when it comes to these things, when there's loads of questions that we have been through today, hopefully that's somewhat helpful. But I can still stick around for a few minutes if anybody has any other specific questions. Seems not. Right, okay, fine. I am going to pause here and return in 10 minutes. Oh, question 8, booklet 2. Okay, well, again, I don't like doing that entire question. I'm just not, I kind of almost refuse. Um, no, it's not an entire question. That's quite a good question. But human skin, groups like we see on the skin, give two ways in which the body protects itself from these microbes. Your skin basically is just in the way, firstly. So it can so if you've got stuff on your skin, what's stopping it from entering? You could mention the fact that the body kills your white blood cells, kill microbes that enter your body. You could mention your stomach producing hydrochloric acid. There's quite a lot of things that your body does. essentially, if the question said, how does your body protect itself from disease? There's loads of answers. Your white blood cells, your hydrochloric acid in the stomach. You could mention your mucus or stuff in your nose. You could mention that when you cut yourself, you form blood clots. These are all answers to the question. Um, right, last one, possibly, depending on what it is. Okay, so this question basically says, yeah, I'm not doing any more questions. We're done. So after Leah should have taken antibiotics for 8 days, she felt much better after 5 days or stopped taking antibiotics. 2 days later, she felt very ill again. Use the graph to explain why. So look, after 5 days, right, let's pretend this is the number of bacteria in your body, right? You took some antibiotics, and after 5 days, you started feeling better. And so you stopped. The problem is there was still some bacteria in your body after 5 days. And that bacteria, let's say there's this much bacteria, what does it do? It multiplies, which means that if you don't finish your course of antibiotics and kill all of the bacteria, it can come back and make you even more ill. So that's why um, that happened, essentially. So we had not killed all of the bacteria, which means it reproduced and it returned and made you more ill. Right, that's enough for that. Remember, general rule for these lessons is try to have a go at the homework before the session begins, and then ask your questions at the beginning. So when you keep firing questions 50 minutes in, it makes me think that you're doing it right now for the first time. So I will return in 8 minutes to assist people with year 10 and year 11 stuff who did disease and ecology. And so we will do some of those questions in a few minutes. Hello. Right. Um, we did a couple of topics with year 10 and year 11. So we did the topic of disease, including everything to do with white blood cells and vaccinations, um, and possibly drug testing that you might have got onto. If you have any questions from the year 10 booklet, please feel free to ask me. Some of these I've done because they were repeated in the earlier one, but I'm happy to do any more. And for year 11, we did a topic called ecology, which is all to do with, oh, there's quite a variety of different things actually in ecology. If you do have any homework questions from any of those, please feel free to ask them. 1D is the first question. Let's have a look at it. Um, right, this one. So, um, in this question, there is a practical experiment done on three different antibiotics, and we tried them on three different bacterias. So we got bacteria A, bacteria B, bacteria C. And what these circles show are, after a little while, how much of that bacteria has been killed by that particular antibiotic. And so what you have to do for question D is compare the three antibiotics. So for example, I would say that antibiotic 1 kills some of bacteria A, but it's most effective against bacteria B because look, we get the biggest circle around bacteria around 1 on this one. And it's quite effective against bacteria C. So it's most effective against B, then quite effective against C, then doesn't really kill much of A at all. So that would be how I would explain number 1. But number 2, we move on to and say number 2 is effective against all three of them, but it's most effective against bacteria C. So we get the biggest circle around 2 here. So it's obviously killed a lot of that bacteria. So if you were a doctor, for example, and somebody has this bacteria C, then you may choose number 2 against it because it seems to be the most effective against bacteria C. And then number 3 is effective against bacteria A. It kills quite a lot of bacteria A, a bit less of bacteria B. It doesn't kill any of bacteria C, so you wouldn't use number 3 against bacteria C. That's how you answer that particular question. The Defina question, I don't know what that means. What does that mean? I mean, I haven't seen these homeworks, so I just look at them there and then. Is there a question number associated with that? Um, are they near the end somewhere? Um, that's fine. Yeah, it hopefully will arrive before Sunday, but if not, don't worry, you can give it the week after. It just takes a while sometimes before it goes out, so don't worry. But you can still um, follow along if you wish. But this is what the homework would have been in terms of that. Um, oh God, why is this happening? Right. Anybody got any other actual questions from the oh, if you tell me a number, then I can go to it. If not, then I will yes, this does get recorded. It ends up on our YouTube thing after a while. Um soon after it ends. 3B on year 10. Let's have a look. If any of these questions are about how a vaccine works, um, give two ways a person with mild infection of salmonella can help reduce the spread of the bacteria to other people. Right, salmonella is essentially food poisoning, and it is caused by a bacteria. That's what it's caused by. Um, and so, what you could do, for example, is make sure you wash your hands um, after going to the toilet, something like that. It's basically related to food, so it's like food that's not cooked properly, or that's how it's spread. Basically, it's through food. So, washing hands after the toilet would probably be a satisfactory answer to that question. Washing hands before you handle food, for example, would work. So not just wash hands on its own. You need to say specifically, like after going to the toilet or after being sick or before you handle food, that kind of thing. In general, if you're ill at all with any disease, then one answer to avoid it being spread is to isolate yourself. If you isolate, there is no way of you passing it on to another person. That's why during COVID we were told to isolate a lot. So that would be a sensible answer to that question. Any other specific questions? Otherwise, I might just do most of this. A pathogen, for example, is a microorganism that enters your body and causes disease. Probably don't need all of that. Oh, one of these, again, in case you didn't come across it in the lesson, MMR is a combination of three diseases called measles, mumps, and rubella. You get the vaccine containing all of them. 16 in the back. Let's have a look at that. Right. Scientists want to make human stem cells from body cells rather than getting stem cells from embryos. This must be a moral thing. Um, So this is not really in this topic, but stem cells are these cells that are special in that they can turn into any other type of cell. Um, we can find them in the body now, in certain places, in bone marrow specifically, or you can get them from embryos. Generally, we don't like getting them from embryos quite so much because, let me see if I've got the answer to this question somewhere. Um, Some people object to destroying human embryos. That it's a moral thing, right? We generally feel that it's wrong to take things from an embryo because it doesn't have a right to say no, kind of thing. And so, yeah, B is the answer to that question. Could you please go over monoclonal antibodies? I can do monoclonal antibodies. Um, yeah, it's quite complicated, monoclonal antibodies, but I will go over that now. Surface area to volume ratio and what it means. Well, yeah, I'll talk about that in a second as well. Okay, assuming there's no other major questions here, but if you do have them, then keep asking them. I'm going to open up some notes on monoclonal antibodies. 1E part onwards, year 10, I assume that means. Um, 1E, 1E, okay. Is that at the beginning? Okay. Very awkward, these numbers. Um, Okay, question one. I'm going to do all of question one because it's quite long. Well, I did that first part already. Milk contains bacteria. Excellent. A small volume of raw milk was placed in a counting chamber in a special type of microscope slide. Figure two shows what the counting chamber looked like when viewed using a microscope. So these are some bacteria. You can see that there. Um, a scientist counted the number of bacteria in four samples of raw milk. So this is the number of bacteria in E, F, G, and H. Which milk sample is shown in figure two? Do we just count? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. I count 15 there, which means that that presumably is E. Um, Calculate the mean number of bacteria in the four samples. This is a maths question. To work out the mean, you add up some numbers and divide by how many there are. So you'd add up those four numbers and divide by four. Um, oh God, okay. Right. So let's follow these steps. Calculate the total area of the counting chamber in figure two. To work out the area of this, you do 0.1 times 0.1, which is 0.01 millimeter squared. Okay. Um, sorry, that's not there. That's here. Apologies. Um, then calculate the volume of the counting chamber. It tells you to go work out volume by doing area times depth. We got area of 0.01 times the depth, which they tell you is also 0.01, which is 0.0. How many is that? Let's get the answer. 0.0001, something like that. Okay. And then calculate the mean number of bacteria per millimeters cubed of milk in the samples. Just fuse this equation. It says the mean number of bacteria from part F, which I never finished, but it was 14, divided by the volume of the counting chamber, which we worked out as being 0.0001. And if you do that division, you get an answer of 140,000. That's how you would do that question. In terms of tests, your tests will be at the end of the month. So if your lesson is on a Sunday, it'll be the 25th. We've just swapped it around a bit. You were technically meant to have it this Sunday, but we have swapped things around a little bit to give you a bit more time. And you should get an email tomorrow with a list of topics, but you kind of know what they are because it's things you've done recently. Like that's simply the answer that you've done with those. Um, it's at the end of the month, so depending on what day you attend, it's kind of around here. Most of you attend on Friday, Saturday, Sunday, so it'll be around here. If you attend on a Saturday, just to be clear, it's on the 24th, not on the 31st. Yes, so for the Thursdays as well, it will be the 22nd most likely, but I will confirm that with you tomorrow. So it's not going to be this week, so no need to panic. Um, okay, there's more for this question. There is. Milk is heated to reduce the number of bacteria it contains before it's sold for humans to drink. Um, Milk with more than 20,000 bacteria cannot be sold for humans to drink. This shows the number of bacteria per centimeter cubed in four different samples. Right, this is a question about standard form. Um, you need to understand what these numbers actually are. So this number, for example, is 18,000. This number is 22,000. This number is like 0.00022 or something. This number is 1,800. The question is, milk with more than 20,000 cannot be sold. The only number that's more than 20,000 is that Q. So it is Q. Why should milk sold for humans not contain large numbers of bacteria? Probably because it can make them ill. Let's see what the answer specifically says. Yeah, it could make humans ill. That's the answer. How is the test done? Is it all science together? Two-part two, I'll do in a second. Question 17, yes, I'll do that in a second. The test is done for those of you, again, that are new. You have a single document. In fact, what I'll do is I'll quickly do this. So this was the test that people did in October. So it is a single document that looks a bit like this. It has a small biology section, a small chemistry section, a small physics section, and a small math section. There you go, all in one. And you also have a verbal test during it, which is good fun, because you get asked some questions face to face. Two-part two. Oh my God, that's all question one. Two-part two. Is that it? This is what you want? Two A part two? What percentage of 16-year-olds are active in the UK? Isn't that just reading off this graph? 60 odd, something like that. And well, we can. If you tell me what question number you want from the homework, I'll switch over to it. You just got to tell me what question number from the ecology homework. Um, the verbal questions can be things like, tell me the definition of photosynthesis. Sorry, the equation of photosynthesis. That could be an equation, that kind of thing. Or it could be based on what we did here with disease. It could be describe how vaccinations work. Off you go, face to face. That's how it works. Um, Bacteria may become resistant. How can doctors reduce the number of bacteria that become resistant to antibiotics? So, bacteria become resistant to antibiotics if we overuse antibiotics, because that's how evolution works. The more you kind of kill them, the more they evolve to become resistant. So, the way doctors can reduce it is avoid over prescribing them. So, or let's put it this way, don't prescribe for minor illnesses. So if you've got a little bit of an illness, you don't need antibiotics because you remember your white blood cells will still fight it to get rid of it. You don't need a medicine. Um, and also, you definitely don't want to give them if someone has a virus because viruses, antibiotics don't do anything to the virus. So you don't want to just give them unnecessarily. That's what you're trying to do. 1D, did I do that already? I did do 1D earlier, did I? Oh no, I didn't. Oh yes, I did. I did 1D. I watched the recording for it. I've done it twice now actually today. It's about comparing these, so you can watch that back later. Um, so yes, if you need any questions from the ecology booklet, then please tell me specifically. And I'll just switch. I've got it here, ready to go. But nobody seems to ask because the year 11s don't seem to show up here as frequently as the other ones. Um, Done for B. Have I done anything else? Oh yeah, question 17, which comes right after question 5 in year 10 booklet. This one. Um, Wow. Okay. This thing is a bacterium that can be found in soil. It causes tetanus. Children are vaccinated against it. Explain why these children do not get tetanus. Again, I'm not going to do that because the question is basically asking you how vaccinations work. Um, and yeah, I mean, it is that is the question. How do vaccinations work? Um, and so we should be comfortable answering that question. So these children don't get tetanus because they've been vaccinated, because when they were vaccinated, they were injected with a dead version of the tetanus bacteria. Their white blood cells produced antibodies, and so they figured out how to destroy the tetanus bacteria. When these children get the real bacteria entering their body, their white blood cells quickly, the word quickly is important here, produce the correct antibody to kill the bacteria. I'm not looking at the mark scheme, but that has got to be something to do with it. I'm not sure if I even have the answer here. Oh, it's here. Yes, so they're immune. The vaccination contained a dead version of it, so they produce antibodies, and so they get a faster response to it. So that sort of thing. Um, 5C. Have I done that already? Is that this one? No, I'm not going to do 5C on the basis that your notes will tell you how a vaccine works. And I did just technically say it as well. Um, so remember, these kind of things, they're in your notes. 100% you have got notes about how a vaccine works, because it is basically the most important part of this topic. I should also warn you, for those of you that are going to do a verbal test on this, I can guarantee that in your verbal test, there is a question asking you how a vaccine makes you immune from disease. Essentially, this question. So you need to say things like, a vaccine contains a dead version of a pathogen. Your white blood cells then produce antibodies to kill it. If you later get the real disease, your white blood cells can quickly produce the correct antibody because they basically remember how they killed it the first time. Ecology 3B. Yep, I'm going to move on to ecology now. I think that's enough of that. Thank you. Good timing. So 3B, let's do this. Okay, right. Quite a big question. And to be fair, you wouldn't have done the required practical, at least in the lesson, because we do a separate lesson on it. Yet it appears in this topic. So let me talk about this entire question first. Some students investigate the size of population of dandelion, please. Right, okay. They wanted to work out roughly how many dandelions there are in this field. They placed a 1 meter by 1 meter square quadrat in 10 random places and they counted the number of dandelion plants in each one. And here are the numbers they got for each one. Why did it, sorry, why did they place it at random positions? They placed it at random positions so they get a more, well, it avoids bias, doesn't it? Because it's in random places. Or you can say it is more accurate, basically, if you do it that way, than just sticking it all in one place. Estimate the total number of dandelion plants in the field. This is how you do it. You need to work out the mean number of dandelion plants here by adding these all up and dividing by how many there are. Without me doing this, what is that? The mean is 6. So that means that on average, there are 6 dandelion plants in one of these squares on average. And this square has an area of 1 meter squared. So in 1 meter squared of this field, we're guessing that on average, there are 6 dandelions. But how big is the whole field? That's where you need to, unfortunately, do some maths and work out the area of this field. This field is, this section of it is 100 by 100, which is 10,000 meters squared. This area is 150 by 300, which is 45,000 meters squared. So the total area of this field is 55,000 meters squared. In 1 meter squared, we basically figured out that there's 6 dandelions. So in 55,000, there is 55,000 times 6, which is 330,000. A lot of dandelions. Um, they want the answer in standard form. 330,000 in standard form is 3.3 times 10 to the power 1, 2, 3, 4, 5. Bit of maths in that as well. I'm probably not going to go back to the year 10 booklet at this point, but we'll see near the end. Um, how many verbal questions will we get? And there's about 3 per science, I think. There's like 3 biology ones, 3 chemistry ones, 3 physics ones, something like that. Right, and that's 3B done, right? Okay, 5F, 5 at the back, and the whole of 7. I probably need to read this question. Just give me 1 second. Let's glance through what's going on here. Because, right, okay, that's obviously a primary consumer pyramid of biomass. Okay, okay, that's all. Hopefully, nobody's asked me that because I think it's not that difficult. Um, a concentration of 5 of fertilizer caused a large increase in the population. I'll definitely explain why. Um, well, because that's a hard question. I'm just trying to think the answers earlier on somewhere here. Uh, so this goes back to this, right? This thing eats algae. That's really important that you understand that. This eats algae. Algae is something that grows on a pond. When fertilizer goes into a pond, it creates quite a lot of algae. It's like the green stuff sometimes you find on the top of a pond. So, as a result of more fertilizer, what that did was it increased the amount of algae in the pond. And algae is food for this thing here, as we learnt ages ago in the question. And therefore, if there's more algae, there will be more food for this Daphnia, which results in its numbers increasing. So that's what you want to recognize here. That's a bit of a leap, but you have to make here to understand that more fertilizer in a pond results in more algae growing on the pond. More algae happens to be the food for this thing. If there's more food for one thing, there's more of the thing that eats it. Um, right, that was five there. Five at the back and the whole of seven. Let's do the whole of seven. Deforestation affects the environment in many ways. Deforestation is the amount of carbon dioxide in the atmosphere. Two reasons why. Deforestation is bad for global warming for two reasons. If you cut down trees, that's what deforestation is, then it reduces the amount of carbon dioxide taken in by those trees for photosynthesis, right? Trees and plants take in um, carbon dioxide. So, and remember, carbon dioxide is a greenhouse gas that adds to global warming. So, if you're cutting down trees that are usually removing the carbon dioxide, then you are now not removing the carbon dioxide. And so it increases the amount of carbon dioxide because there would be less photosynthesis happening because you're cutting down the trees. Secondly, what we normally do with these trees, because the reason we're cutting down the trees generally is because we are trying to make space for building whatever or farming or whatever it might be. That's usually why we're cutting down the trees. We're not cutting down the trees because we suddenly want loads of wood, which is a common misconception. There's not like a shortage of pencils that we're trying to cut all the trees down for. It is mainly to make space to then grow food or to build houses or whatever because there's more people on Earth, right? Therefore, what we generally do with those trees when we cut them down is we burn them. And unfortunately, burning those trees also increases the amount of carbon dioxide. So, so there's a double effect of cutting down trees. Firstly, those trees are no longer taking in carbon dioxide, and secondly, we're burning them, which is then adding carbon dioxide. So it's bad. It's really bad. What is meant by biodiversity? The exact answer to that question is the range of different species or the variety of different plants and animals on Earth. That's what biodiversity is. Two reasons why it's important to prevent organisms becoming extinct. Um, So there is usually a food chain effect if one organism becomes extinct. Because if something becomes extinct, it's usually food for another organism. So you can say something like it disrupts the food chain. So if one thing becomes extinct, it's usually food for something else, and therefore that thing will be affected if something becomes extinct. Um, and we could also have like a moral answer to this, which is that we should preserve things for future generations. You could say something like that. You could also say that some organisms produce substances that might be useful to humans, like a lot of plants will produce certain useful chemicals and so on. Um, so they may produce substances useful to humans. Or as I say, you could put like a moral spin on it, which is that we should be protecting organisms for the future generations and stuff like that, rather than just killing them all. Um, oh wait, hang on, let me read this. If plants get cut off, they release CO2 that they had stored. Well, no, you just if you kill if you what's the word I'm trying to say? If you just because you cut a plant doesn't mean they release CO2. Like when you cut your grass outside, you're not releasing loads of CO2. CO2 only gets released if they then subsequently get burned, for example. The less photosynthesis is obviously correct, but you need to mention the burning. So you just cutting a plant doesn't release CO2. Can you just say reduces biodiversity? Um, Let me see. Yeah, I would mark it correct. Um, because I think that basically is very similar to this idea. Um, yeah. And it's very similar to that whole idea of the future generations, if there's more biodiversity and stuff like that. So yeah, I would probably say that that's okay. Um, that was all of question seven. And then there was another question relating to um, five at the back. Most food chains have a maximum of five trophic levels. That's the sort of levels of different things in there. Um, and the answer is B. Essentially, if I've got, let's do a little quick food chain. Grass, rabbit, fox. Okay. This grass takes in lots of energy, or has lots of energy because it gets its energy from the sun. Okay. Um, let's just use some numbers here. Let's say 1,000 joules of energy, just for the sake of it, right? Some rabbits eat the grass, okay? But the amount of energy that's passed up a food chain is always decreasing because a lot of that energy is used by the grass to grow or to reproduce or whatever it might be. So its energy is being lost at each level. Okay. Then when the fox eats the rabbit, again, not all 700 is being transferred to the fox. It might be like 300 is being transferred to the fox because the rabbit again is using a lot of that energy throughout its life to grow, to move, and so on. And also, there's one really obvious answer here. When a fox eats a rabbit, it tends not to eat all of it. Um, like it will, like bones, for example, we don't eat. And foxes don't necessarily eat the bones of a rabbit. And yet, a lot of the energy is still contained within those things as well. So that's why energy is lost as you sort of transfer up a food chain. And if you just continue this food chain, then that number is going to get less and less and less. So there's a limit to how many levels there can be to this, because at the end, there's just nothing left. And so the answer is that energy is lost at each trophic level. Let me see what else it says. Um, did you do the questions above? I didn't have time to look at them. No, I only do questions that are asked. So I'm not just doing an entire booklet. I'm basing it on the questions that are asked based on your attempts at doing a question. But I can just skim through some of these, just have a look at what else there is here. Um, deforestation has decreased the amount of forest cover on the island. What's going on here? Describe the change in pattern of forest cover on the island. Is it just that it's decreased clearly. Um, um, and certainly at the edges, it seems to have been removed quite a lot. Um, like, for example, quite a lot of it on the east and the southeast has been removed. You want to specify an area, a part of it, if you want. Um, oh, sorry, there was another question that somebody asked from the one before. See if I can get there. 5D and E. I'll do that. Oh, I did that. Oh no, I didn't. Calculate the mean number. Oh God, right. Okay, that's tricky. So firstly, let's work out the mean of these numbers by adding them all up and dividing by 5. 5 plus 21 plus 16 plus 28 is 70 divided by 5 is 14. So 14 is the mean here. So that means what this is telling you is that on average, there are 14 of these Daphnia in 1 dm cubed. Okay. Okay. But we want to know the number in 1 meters cubed. Now, 1 meters cubed, as it tells you here, is 1,000 of these. So 1 meter cubed as an area is 1,000 times bigger than a dm cubed. Um, which means that if there's 14 in one of those, there would be 14,000 in 1 meters cubed. So that is the answer to that question. Just making sure that is correct. Yeah, it is 14,000. Um, and then E, the pond was a rectangular shape. It's unusual. Measuring 2.5 meters by 1.5 meters by 0.5 meters. Calculate the estimated number of daphne in the pond. So we know that in 1 meters cubed, there are 14,000 of these because that's what we did. We worked out earlier. We've got this pond, which has a volume of 2.5 times 1.5 times 0.5. That's how you work out the volume here. That is 1.875 meters cubed. Make sure I'm doing this right. Um, yes, it is. And so, if I know that there's this many in 1 meters cubed, if I times this by this, it will tell me how many are in this pond because this pond is bigger than 1 meters cubed. So you times that by 14,000 and you would reach your answer to this question. Which is 26,250. How many there are there? Don't you have to discard the anomalies for the mean? Whew, a good question. Um, you see, in this case, that isn't an anomaly. Um, or is it? Wait. Um, let me have a look. I agree with you to an extent. Um, it's not quite the same, right? So, I mean, I don't know. How do you know what's anomalous here? This is the problem here. I don't think there is any anomalies here. This would, I mean, I presume you're saying zero might be an anomaly or five might be, but they're quite spread out, these numbers. So like 28, 21, 16, 0, and 5. It's not like I would argue that if you had 0, 28, 27, 27, 28, then that might be anomalous. I don't see an anomalous result in here. And so no is the answer. I think it's just because there's a zero doesn't mean it's anomalous. Because there's another one here that's five. To me, that suggests that that's not anomalous. Because there's another one that is five. Anomalous is very much unique on its own, that's miles away from everything else. And that isn't the case here. I didn't include, yeah. So firstly, but that doesn't make sense either, because to me then so you discarded five and zero, but then I would argue, why did you include 16? Because 21 and 28, like, yeah, I can understand your point, but I'm just looking at the mark scheme and it isn't. And I wouldn't have discarded here because they kind of is a very specific situation when all of the numbers are very close together and like one of them is miles away. That's an anomalous result. I don't think there is an argument that any of these are anomalous. Um, so let me move on to this topic of monoclonal antibodies, which is all I want to do is just read through some notes of mine, which I have somewhere. Um, you know what? There was another question earlier. Hang on, let me just check if there was something else. 1G in year 10. Let me just see if I did that already. Yes, I did. So I've explained that in an earlier one, so you can watch it back later. Um, oh, you know what? I don't. Let me check the chemistry questions that you just said. Um, oh, right. Oh, God. Okay. I will reply to that chemistry question separately, if that's all right, because I've literally just looked at it. Right. Let me just briefly talk through some monoclonal antibodies. Okay. Um, Monoclonal antibodies. Firstly, let me give a tiny bit of background to what this is. It is quite complicated to get your head around, and it's unlikely to be understood in a short amount of time. But firstly, um, what I will do as well, actually, I will open up my other notes on this. Okay. What we need to understand is every chemical, um, has something called an antigen, which is a unique shape around its outside. Think of it as like a barcode. That's like its unique identifier. It tells you what it is exactly. Every chemical has that. Now, we talk about antigens in the context of bacteria and viruses. Yes, they do have antigens, but so does basically everything. That's important. Like everything has an antigen. Um, and so in your body, when you get ill, your white blood cells produce these things called antibodies, which fit onto that particular shape for the virus or bacteria that it might be. But as I said, because every chemical has this shape or has a unique shape, we can find an antibody to fit that. So, for example, if I had some other chemical, right, and it had these shapes around the outside, okay, then what I could do is there could be an antibody that fits that particular shape. Okay. So it would lock onto it essentially perfectly. Right. And that brings us to the background of what monoclonal antibodies are. They are antibodies that we have specifically made to fit onto a specific chemical. As I said, your body naturally produces antibodies for viruses and bacteria and so on, but monoclonal antibodies are what we kind of try to make for any particular chemical. Let me jump to the end first and explain one of the uses. So I think you have asked a question about um, how to make them for certain diseases. There you go. I'm about to do that. So what we are looking at here, if we just look at this picture here, are some cells in someone's body. So the blue ones here are essentially normal cells. And you see these little triangles? These are those antigens that I mentioned. So every single cell basically has these unique markers around the outside. Um, and then on the right here, these are cancer cells. So this person has cancer. Cancer cells also have these antigens, and these are different, as you can see, to the normal body cell markers. Okay. Um, and so, if it were possible to make an antibody that only attaches onto this, then we could do something useful. Okay, with that. And that's what we try to do. So we have made some antibodies, monoclonal antibodies, that only fit on, as you can see here, to those cells. Okay, they don't fit on to these ones, so they don't go to your normal body cells. They only attach on to the cancer cells. And what we also can do is we can attach certain drugs to antibodies. These drugs can kill cells, but they will only kill the cell that the antibody attaches to. So these antibodies will not attach to these normal body cells. They will only attach to these cancer cells because they've been specifically made to attach to those cancer cells. That is how we can make antibodies. Well, that is how we can use them to treat certain diseases, basically. So we can make an antibody specific that fits onto the antigen of that disease. Okay. But the question is, how do we make them? So, and the answer is we use some mice. Right. Let's say that I've got this cancer cell and it has these sort of markers on the outside. These are the antigens. Right. What we do is we want to produce antibodies that fit onto that shape. That's what we want to happen. Um, and we then inject a mouse with that antigen. So, in other words, that shape that for which we want antibodies. The mouse's body does what our body would do. The white blood cells, oops, the white blood cells start to produce antibodies that fit onto this shape because that's what our body does, right? When you have a virus or a bacteria in your body, we start to produce these antibodies. So the mouse's body starts to produce them, or the white blood cells start to produce them. We then remove those white blood cells. So these white blood cells are special in that they only produce the antibody that locks on to the antigen that we injected into the mouse. The problem with white blood cells is that they don't really multiply easily. So ideally, we want lots of these white blood cells because we kind of want lots of those antibodies, but we've literally only got like one or two. We want lots of them. So there's this extra step in this topic of monoclonal antibodies that we get a tumor cell. A tumor cell is one that multiplies really quickly. Tumor is basically like a cancer cell. It multiplies really, really quickly. We then fuse a tumor cell with those white blood cells that we took from the mouse. And we get something called a hybridomer, which is a mix of this white blood cell and this tumor cell. So remember, this white blood cell produces these antibodies that fit onto the antigen that we injected into the mouse. But now that we've fused it with a tumor cell, this will not only produce the antibodies, but it will also multiply. So we will get lots and lots of these white blood cells that produce the monoclonal antibodies that we want. And that is how we have produced an antibody that fits on to whatever we want it to fit on, because the thing that we wanted it to fit on, we injected into the mouse to begin with. So the mouse's white blood cells started making them, and that led to us having monoclonal antibodies. There is an example of monoclonal antibodies to do with pregnancy tests, which I'm not going to go into right now, but that is the kind of one where it's, you need to know that in a bit of detail to how that works. Right, last thing I want to do then, give me an idea of 5C ecology. I can try. I thought I did that. How do I do that? Oh, this one. Give a reason why the total biomass of the Daphne in the pond is different from the total biomass of the algae. Um, Probably because they didn't eat all of it. Yeah, that's essentially the answer. Right. So essentially what we're saying, yeah. So we have some algae. Imagine this algae, right? It has a certain biomass. Let's pretend, use a fake number, 1,000. Right. We have these Daphne. I don't even know what these Daphne are. What are they? Anyway, we'll pretend they're fish. Okay. Right. They eat this stuff, right? And so, but they don't eat all of it, do they? So the total biomass of this is likely to be less than the total biomass of this. Um, and that's it. That's the answer. That's one answer. There's a few other answers that you can lose it in respiration and stuff like that. But I think the simplest answer is that not all of the algae gets eaten. Is that okay? No. 3C. Inspiring questions, aren't we? Oh, geez. Right. So we talked about this required practical, which was about measuring the or estimating how many dandelions there are in a field. For this question, without me doing it all, right? I'm not going to do it all, but the question is feces. Yes, you can mention feces as well, indeed, in the last question. Um, right. Quadrats 5, 7, and 8 will each place less than 10 meters from the woodland. These quadrats contain low numbers. Light intensity affects the number of dandelion plants that grow in an area. Plan and investigate, test the hypothesis. So what you might do is, so find, you're trying to figure out whether you get fewer dandelions in areas of low light versus areas with a lot of light. So what I would do if I was investigating this, I would create an area that's in the shade, so to speak, right? So where there's less light intensity, and randomly throw, let's say, 4 or 5 quadrats in that area and work out the average or the mean number of dandelions that are in that area. Okay. Then you would pick an area where the light is greater, like let's say further away from those trees. Randomly throw some quadrats in that area and work out the mean. And then what you can do is compare these two means. So you have an average for the number of dandelions that are in the sort of dark, okay, um, and or at low light intensity. And then you can see the average number of dandelions where there is a high light intensity, which is further away from the trees, for example. And then you compare the two. That's how you plan the investigation. You are going to basically imagine you are doing the experiment to figure out whether or not light affects the growth of it. And so, yeah, you want to put a lot of quadrats in random places, some, though, in areas with lots of light and some with areas in less light. For each quadrat, count the number of dandelions and calculate a mean, and then compare the data from the different light intensities. So see if there's more where there's more light and less where there's less light. That's how you would go about doing that. Right, I think that's sufficient, hopefully, for today. This weekend, you're supposed to be doing some maths, even though you're meant to be doing chemistry, but you're not. So just to let you know, it will be some maths this weekend. And then we will do some maths next week as well. If you have any other questions, I will, for the person that sent the question on the WhatsApp, I will try and get back to you later today or tomorrow. And that's it, I think, for today. I'll let you go. I want to go.